Thank you very much. So I took the difficult task to give you an update on the therapeutics development for CMT. It's difficult, but it's also a good time for CMT therapies because there are so many developments, and I can only highlight a few. Apologies to those whom the work I will not show, but I think you will appreciate that there is a lot going on, and hopefully in the near future we will have treatments also available to patients. So as we all know, we have a long path from the conception, the proof of concept in vitro, in animal models, in big animal testing, to clinical trials, and then to regulatory approvals. So where do we stand today in terms of CMT therapies? For sure, drug therapies are much more advanced. So we have several examples that reached clinical trials, including the PXT, completed the short neuropathy you heard yesterday from Applied Therapeutics, the L-serine trial run by Mary Riley's group. HDAC6 inhibitors are in the first in-human trial, and the chloride channel inhibitors. And soon we will see also the TRPV4 channel inhibitors, and hopefully also other drugs like the GCN2 inhibitor from Alesta Therapeutics. To treat the cause of CMT neuropathies, we need more gene therapies, and they are not so advanced. We have one that is in the clinic, the CMT2S gene replacement, which has been ongoing. I don't have any update on that to give you. There are some other programs that are nearing the clinic, especially for CMT1A, 1X, and 4J, and some other programs that are still at the stage of the rodent model testing. And of course, as I said, we still don't have any approved therapies, so we still all have a lot of work to do. So I will borrow this nice figure from the recent review from Davide's group and De Grado, showing you the interplay between the neuron and the Schwann cell, and how many different ways there is to disturb this fascinating and highly differentiated myelinated nerve. And this gives us also a lot of therapeutic targets and a lot of genes to work with. And I try here to summarize the pharmacological targets first. The ones that reach clinical trials are especially the aldose reductase inhibitor for the short CMT. It's a metabolic neuropathy, so it's really amenable to a drug therapy to reduce toxic sorbitol levels. It does, yes. Then we have another specific type, the L-serine, to reduce toxic deoxy sphingolipid levels in the HSN1. And then we have a non-specific approach, the chloride channel 1 in the muscle, to hopefully help boost the function in different CMT types. Still at the preclinical level, we have the HDAC6 inhibitors. So this is, I will have a few slides on that. This is supposed to rescue axonal degeneration and will be applicable to multiple CMTs. Then we have also the CEFIN1 story to reduce the ER stress from Maurizio De Antonio's lab, and will be possibly useful for CMT1A and 1B. We have the neuregulin pathways that regulate myelin thickness, and this is work from Ale Bolino's lab, and niacin, niaspan would be good for some CMT types with either reduced myelin formation or excessive myelin formation. Then we have yes, we have. Okay, so the next one, another line of research has been focusing on inflammation. That's work from Rudolf Martini's lab, the colony-stimulating factor receptor inhibitors, especially shown for CMT1X and 1B. And then we also have another story on SARM1 inhibitors to block axon degeneration. And I will have a slide on this because it's very intriguing. The TRPV4 inhibitors for CMT2C, And finally, the tRNA synthetase CMTs, we have some treatment to treat the integrated stress response with GCN2 inhibitor. So I will show you some example of this. It's not very responsive, like the outcome measures we have for CMTs. So, yes, good. Okay, so SARM1 targeting and which CMTs. So I'm not going to go into too much details of the biochemistry, but SARM1 seems to be a master regulator of axonal degeneration. Is the final common pathway that is triggered by an imbalance between NMN and NAD, triggered by chemotherapy, by toxins, or by genetic mutations. So there was evidence from chemo and diabetic-induced neuropathy that you can rescue the axonal degeneration with SARM1 inhibitors. The first promise for CMT came from CMT2A model. So when you delete the SARM1, you improve the phenotype in the CMT2A model. But then we had a series of other papers showing that SARM1 inhibition does not work for CMT1A in the model, and recently just published also in 3 models of other CMT2s, the 2D, the 2E, and the 2S. So it was not useful reducing the level or inhibiting SARM1 in these types. So we conclude that SARM1 inhibition is unlikely to be a magic bullet for many CMTs, but may be useful at least for CMT2A, and maybe we have to take a more differentiated approach and look for the biochemical imbalance and whether indeed SARM1 level is upregulated in certain CMT types that will be amenable to inhibition. So it's story to be continued. The other story that is exciting and comes from Ludo van der Bosch lab is the histone deacetylase 6 inhibition. So HDAC6 is responsible for deacetylating alpha tubulin, a major component of axonal microtubules responsible for axonal transport and in general integrity. So it has been shown that HDAC6 affects the degradation of toxic proteins and also the integrity of the axonal cytoskeleton. So deacetylation of tubulin will disrupt axonal transport and even has a role in regeneration of the axon at the axon terminal. So it was shown in different models, initially in HSPP1 mice, that if you inhibit the HDAC6, you can improve the phenotype of these CMT types, and then in further models, in CMT2A, CMT2D, and also in CMT1A more recently, there was a reduction in the aggregates and improvement of function. So this approach is now being taken to the clinic by Augustine Therapeutics. So there was an exciting development with the first patient dosed last May. It's a selective HDAC6 inhibitor, and I took this from the company's website. So it's supposed to be have a good biodistribution to the nerve and be stable and be safe. So we look forward to seeing the further results of this phase 1 trial. The next approach that I want to go over briefly is the chloride, the CLC1 inhibition. This is work that is done by NMD Pharma. So this is the major ion channel on the muscle membrane that is responsible for repolarizing the muscle membrane after the excitation. So actually, loss of function of this channel causes myotonia, muscle stiffness. So here, the idea is that in CMT patients, it was shown that there is a defect in neuromuscular transmission. So single fiber EMG shows increased jitter and blocking. And this NMJ dysfunction correlates with other functional outcomes. So inhibition of the chloride channel in 2 CMT models including the CMT1A and CMT2D mice, resulted in improved muscle strength and improved electrophysiological measures. So this may be something that could benefit different CMT types, regardless of the etiology, by boosting neuromuscular transmission. And is also currently in clinical trial. There was one short trial completed, but there is a plan for a longer trial as well. I'm moving to the next. Very briefly, a few things about the CMT2C treatment, the TRPV4. So this is a channel, it's a calcium channel, and it was associated with CMT2C through gain-of-function mutations. That was work in Charlotte Sumner's lab. So the mutations that cause CMT2C actually result in prolonged channel opening, causing toxic gain-of-function effects. And recently, work from Sullivan et al. Showed that in the model of the disease, there is, if you knock in the mutant channels, you can reproduce the phenotype. And then if you delete completely the channel, you actually rescue the phenotype, clearly supporting a gain-of-function mechanism. And further work showed that there is a leakage in the blood-spinal cord barrier, especially shown here to affect motor neurons. And that's probably what's driving the pathology. And interestingly, this is a disease that presents with vocal cord paralysis and not necessarily length-dependent involvement. And so this therapy now, it's an inhibitor for the TRPV4 channel that is being developed by Axonal Therapeutics, if the next slide comes up. So by inhibiting the channel pharmacologically, it was shown that you can actually restore the blood-spinal cord barrier disruption, improve the phenotype in the disease model. And so there is now plan by Axonal to move into a first in-human trial for this disease. So the last pharmacological approach that I would talk about briefly is the one related to tRNA synthetase associated peripheral neuropathies. So this is a group of rare axonal CMTs, CMT2D being the most common, with a glycyl tRNA synthetase, GARS mutation. So these proteins load amino acids to the respective tRNAs for protein translation. And in the patients who have mutations in these tRNAs synthetases, there is a failure of the synthetase to release the tRNA, so that blocks the protein translation. There is work from Rob Burgess' lab and Eric Storkenbaum showing that that initiates an integrated stress response in the cell regulated by GCN2 and downstream by ATF4. So that is clearly a toxic gain of function related to tRNA sequestration and depletion. So there are different approaches to treat this. The first one was by inhibiting GCN2. So if it seems that the integrated stress response is particularly evident in motor neurons in the spinal cord, explaining also the phenotype. And if you pharmacologically or genetically inhibit GCN2, you can actually correct the phenotype. So that was the first approach. And that is now being developed for clinical translation by Alesta Therapeutics. Again, I take this from the website. And I believe there is a plan to move into a clinical trial using the GCN2 inhibitor, and that would be relevant for all tRNA synthetase CMTs. The other approach for this group of diseases is to actually replace the tRNA, and this is work from Eric Storkenbaum's lab. So here, because the tRNA is sequestered and cannot be released, you can actually replace it in the cell. they showed that by delivering using an AAV9 viral vector, the glycine tRNA, they can actually restore the phenotype in the disease model. And so now this is technology being further developed by XtRNA, spin-off from that lab, to lead the clinical translation. And that brings me to the gene therapy approaches that I will go over very briefly. First of all, for the axonal CMTs, so obviously for this we need to target neurons, motor neurons and sensory neurons. So there are different delivery approaches, but it seems that intrathecal approach is the most efficient to really get to the target cells. Different AAV vectors have shown already good promise for this, and so this is a relatively straightforward approach. There are some new viral capsids that seem to be very efficient for targeting neurons, recently published, including the PHPN and the transferrin receptor targeting capsids. And we already have some examples. The first one is CMT2A, the obvious most common axonal CMT type, which is caused by mutations in the mitofusin 2 gene, resulting in mitochondrial mobility defect and energy loss. So initially it was shown that overexpression of mitofusin 1 or mitofusin 2 wild type alleles can actually rescue the phenotype. That was work from Bob Balow lab. And then we also showed in vitro that by overexpressing mitofusin 1, we can actually reduce the aggregation of mitochondria. And there is some work right after my talk that you will hear from Natalie related to this approach. And I will just briefly mention the work from Stefania Gorti's lab in Milan. They use silencing and replacement simultaneously, so knockdown of the mutant mitofusin 2 and then replacement with a wild type allele. This was shown in culture and in vivo, but of course there is a long way to demonstrate this, and there is a lot of work with allele-specific silencing ongoing in different labs. One gene therapy approach that is in the clinic is the CMT2S replacement, the IgMBP2 gene replacement. There is an ongoing clinical trial. It was shown in the mouse models that it can rescue the phenotype. The effect is dose-dependent, and in fact, if you give too much of it, the effect is not as good. So we have to be very careful with gene replacements in neurons to give the right amount of the gene. And so we look forward to further outcomes of this study. The other gene therapy targeting neurons that is close to the clinic is the CMT4J gene replacement for FIG4. So this was shown also to benefit the disease model, and now it is just went through IND approval by FDA, by Albida Therapeutics, and hopefully will move to the clinic as the first gene replacement therapy also for 4J. I will say a few things about Schwann cell targeting gene therapy. So here, obviously, the challenge is a little bit bigger because Schwann cells are all over and along peripheral nerves. So the biodistribution and the delivery becomes more of an issue here. It's not as easy as targeting neurons. We need to get to Schwann cells. And there are, again, different delivery methods. In my lab, we have focused mainly on intrathecal delivery because it requires lower doses and appears to be more efficient. And then we need, doesn't go back, we need also viral vectors that will target Schwann cells. So we have some already validated, but new ones are coming. And I had it in that slide, but anyway, I will move forward. So CMT1A treatment is the one that attracts most attention, of course, because it is the most common. So the idea here is to reduce the overexpression of PMP22, and there are non-viral methods. The one is the fatty acid ligand conjugated, the Falcon technology, and the other one is the squalene nanoparticle from Lilian Massad's group, and both are ongoing in development. And we also had some viral vector approaches. And the goal, of course, is to bring the PMP22 levels close to the physiological of around 100% and not over silence. So I will show you. I show this slide for the last few years because there is nothing new to say about it. So this is the DTX Pharma. So there is a proof of concept. It's still under development by Novartis, and hopefully in the near future there will be a clinical trial for this. And then the squalene nanoparticle story is developing. There was some proof of concept in the mouse model delivering siRNA, and there was also some initial work in non-human primates, and I think this is now ongoing for further validation. We used a vectorized approach to deliver an artificial microRNA packaged into the AAV9 vector, and showed that we get widespread distribution and specific silencing of PMP22 in PNS tissues. And then we did further validation, so this is published work, but then we also did a dose escalation study in the mouse model and showed that there is a dose-dependent improvement in various parameters, including morphology, blood biomarkers, and electrophysiology. We also did our RNA scope analysis with dual detection of the MIR and the PMP22, showing a dose-dependent silencing which was widespread in Schwann cells in the sciatic nerve without causing any excessive silencing of the PMP22. This has also now completed a non-human primate study, scaling up the doses from the mice to the primates with extensive safety and biodistribution analysis. There were no safety signals, and I will just show you the biodistribution data. In the peripheral nerves, there was a widespread biodistribution of the vector after intrathecal injection in the primates. And we also had the target engagement at the mRNA and at the protein level of about a silencing of about 30 to 50%, both in the proximal and in the distal sciatic nerves. So very briefly, I will say a few things about the CMT1X program. CMT1X is the second most common and results from loss of function of connexin 32. So here we want to replace the gene specifically in Schwann cells. We use a cell-specific promoter, the myelin protein 0 promoter. We showed that delivering this with AAV9 in different models of the disease, we can improve the function and the morphology. And we also performed a trial using an AAV-RH10, so a different capsid, in a dose escalation study recently published, confirming these results. Recently, we also completed a non-human primate testing of the gene therapy for CMT1X, delivering the therapeutic gene, showing again an adequate targeting of PNS tissues with intrathecal injection without any toxic effects. So hopefully this will move towards further translation in the near future. And I think I have a couple more minutes. So in terms of the CMT4C, is another loss of function demyelinating CMT type. This is actually the most common recessive demyelinating CMT type caused by loss of function mutations in the SH3 TC2. It's an early childhood and more severely progressive disease. We developed, again here, a gene replacement strategy using a minimal version of the myelin protein 0 promoter to allow packaging into the AAV9 capsid. And we demonstrated a proof of concept beneficial effect in the mouse model of the disease. We continued here also with an updated, more clinically translatable plasmid in a dose escalation study with a human promoter and to deliver the gene. And we showed a dose-dependent improvement in the morphology and in the behavioral function in this mouse model of the disease. And this is the morphology of the femoral nerves. So this has actually gone through pre-IND application to FDA last year in collaboration with UT Southwestern in Dallas. We have a good feedback from FDA, and now is in the process of going through the final GLP toxicity study, and hopefully be available for translation in the near future. So I will close by summarizing, if it responds. Yes. So just summarizing the gene therapy approaches. So we have, and I didn't have time to talk about all of them. We have disease-specific approaches and non-specific ones. The disease-specific, I classify them into axonal therapies. They need to be delivered to neurons, whether it's a gene replacement or a gene editing or silencing or CRISPR, and you have seen some examples. For the demyelinating ones, we have to deliver the treatments to Schwann cells. There are many different approaches for CMT1A and for some other CMT types, mostly gene replacement. And we also have some non-specific approaches with trophic factors. Some of them have been also in clinical testing, like the NT3 and SARM1 and BDNF. So they may be complementary as a possible treatment as well. And with this, thank you, and I would like to thank all the collaborators and all the funding agencies and the patient associations who supported the work in my lab. Thank you.
So thank you very much, Kleopas. You have been perfectly on time, so it's time for questions. Are there questions? Otherwise, maybe you have one? No, I can ask a question then. When you talk about myeloregulating proteins, there is also a fine-tuning between them. So PMP22 to P0, P0 to MBP, and so on and so forth. Do you think that we will be able really to keep this fine-tuning by interfering with the genetic expression of the single myeloregulated proteins?
Well, if we're treating the cause of that specific neuropathy, I think we can. We have seen that the cells have a lot of capacity to auto-regulate also. We have seen the example with PMP22 silencing, which was quite intriguing for us because when we increase the dose of the silencing vector, actually the silencing is less effective. So it seems that the Schwann cells have a way to counteract any excessive silencing. And I was just talking to Lilian, and she sees the same effect with the squalene nanoparticles. So I think Schwann cells have a capacity, and you're right, there is a very fine regulation of the myelin proteins. But if you correct, at least to some degree, the cause of the demyelination, I think the cell will do the rest. As long as we can target the cells, and of course, we all know that patient nerves are not the same as the mouse nerves. Schwann cells are at different stages of pathology, but we have seen that Schwann cells have the potential to regenerate. Axons don't regenerate so easily, so that's the other challenge.